May I have your attention, please? Our session is about to begin. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning and welcome to the fourth International Conference on Biology Education, Natural Science, and Technology, INCOBEST 2025. With the theme, Emerging Issues on Natural Science, Environment, and its Learning Innovation. I am Gendis Anggista, your host for today. It's an honor to me to introduce and welcome the Rektor of Universitas Muhammadiyah Surakarta, Profesor Dr. Harun Joko Prayitno, M.Hum, the Dean of Faculty of Teacher Training and Education, Profesor Dr. Anam Sutopo, M.Hum, the Chairwoman of International Conference on Biology Education, Natural Science, and Technology, INCOBEST 2026, Dr. Efri Roziati, Magister Science. Our keynote speakers, Professor Mathew Seymour, Ph.D. from Hong Kong University, Professor Dr. Insinyur Syafi Samaun, STDEA from King Abdul Aziz University, Mr. Muhammad Lutfi Hidayat, Ph.D. from Universitas Muhammadiyah Surakarta, lecturers and staff of Biology Education Department, and all the participants of the conference. Alhamdulillahirabbil alamin. Thanks to Allah, the Lord of the universe, for His blessing and mercy for us to be gathered here in the fourth International Conference on Biology Education, Natural Science, and Technology, INCOBEST 2026. May salawat and salam be showered down to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us from the darkness of Jahiliyah to the brightness of Islam. Ladies and gentlemen, allow me to read the agenda for today. First, opening session. Second, singing the Indonesian National Anthem and Sang Surya. Third, Al-Quran recitation and interpretation. Fourth, welcoming and opening remarks. Fifth, main event. Sixth, announcement. The last, closing session. Well, ladies and gents, we are now holding the first agenda, the opening by reciting Basmalah together. Bismillahirrahmanirrahim. The second agenda is singing Indonesian National Anthem, Indonesia Raya, and also Muhammadiyah Official Anthem, Sang Surya. I would like to ask all participants to stand up as a tribute to our national anthem.
للإيمان أن آمنوا بربكم فآمنا ربنا فاغفر لنا ذنوبنا وكفر عنا سيئاتنا وتوفنا مع الأبرار اللهم ارحمنا بالقرآن.
In the name of Allah, the most gracious, the most merciful. Indeed, in the creation of the heavens and the earth, and the alternation of the night and the day, are signs for those of understanding, who remember Allah while standing or sitting, or lying on their sides, and give thought to the creation of the heavens and the earth, saying, Our Lord, you did not create this in vain. Exalted are you. Then protect us from the punishment of the fire. Our Lord, indeed whoever you admit to the fire, you have disgraced him. And for the wrongdoers, there are no helpers. Our Lord, we have heard the caller to true belief, proclaiming, Believe in your Lord alone, so we believed. Our Lord, forgive our sins, absolve us for our misdeeds, and cause us to die among the righteous. Allah Almighty has spoken the truth. Wassalamualaikum warahmatullahi wabarakatuh.
Righteous God for all his word. Moving to the fourth agenda is welcoming and opening remarks. The welcoming remark will be delivered to you by the Dean of Faculty of Teacher Training and Education. Please welcome Professor Dr. Anam Sutopo M.Hum.
Okay, thank you, Ms. MC. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Innal hamdalillah nasta'inuhu wa nastaghfiruhu wa na'udzu billahi min syururi anfusina wa min sayyiati a'malina. May yahdihillahu fala mudilla lahu wa man yudlil fala hadiya lahu. Asyhadu an la ilaha illallah wahdahu la syarika lahu wa asyhadu anna Muhammadan abduhu wa rasuluh. Allahumma shalli ala Muhammad wa ala alihi wa sahbihi ajmain, amma ba'du. The Honorable, our Rector, our special person in this room, check sound. Is it okay, my son?
Yes, Prof. Is it okay?
Yeah. Once more, the Honorable, the keynote speaker, Mr. Professor Mathew Seymour, Ph.D. from the University of Hong Kong, Professor Dr. Insinyur Shafi'i Shauman from King Abdul Aziz University, and also my college, and also as the keynote speaker, Mr. Muhammad Lutfi Ph.D. from English Education Study Program, Education Faculty of Universitas Muhammadiyah Surakarta. First of all, and all thanks and praise is due to Allah, the most merciful, the most merciful. Thank you for His mercy and blessing in giving us this opportunity to gather in this excellent forum in line with the International Conference on Biology, or we call this INCOBEST, International Conference on Biology Education, Natural Science and Technology. As the Dean of Education Faculty, let me welcome you all and give the appreciation for conducting this conference. But as Muslim, of course, may peace and blessing be upon to our Prophet Muhammad sallallahu alaihi wasallam, also upon his family, his companions, and his followers until the end of the time. As Muhammadiyah followers, Muhammadiyah members, let's declare our commitment that Ya Allah Ya Rabbiku, Muhammad junjunganku, Al-Islam agamaku, Muhammadiyah gerakanku. And also as the Dean of Education Faculty of Universitas Muhammadiyah Surakarta, I take a great pride to be here, to proud to say congratulations for you all in conducting this international conference. I think this fourth international conference on biology and education, natural science and technology, or INCOBEST, addresses the challenges to bringing together educators, researchers, technologists, and also policymakers under the theme of emerging issues in natural sciences, the environment, and learning innovation. The conference aims to explore recent science advance, examine their educational application, and also share innovative pedagogical practices. And also I think this INCOBEST, this conference provide some strategies and platform for knowledge exchange, collaboration, and also the promotion of innovative learning approaches to prepare a future, and of course to promotion innovation learning approaches to prepare future generation for involving scientific leaders. I think my deep thanks also goes to all committee, especially the head of biology education study program, to Ms. Triastuti, Dr. Triastuti, and also friends committee that successful in conducting this international conference. I think collaboration, partnership are watchword and future development through this opportunity, and it is possible to make our different point of view, to make a collaboration in from all participants. I do hope that this international conference will provide a national and international or global networking to gear up our development of science, especially in biology, education, and also in natural science and technology. Of course, this conference conducted in this opportunity and taking great care as the fundamental and application aspect. And I also think that it will give helpful and valuable further development in our university, in our career, in our country, and also in around the world. I do expect that this international conference will initiate, develop, and also promote some platform that support in line with education, science, education, natural sciences and also technology, innovative ideas and research outcome from all paper presented in this international conference are expected to be a significant milestone of advanced science, technology, and humanity. So once more, it is my great pleasure to welcome you all in our faculty of education, Universitas Muhammadiyah Surakarta. And I hope you all have an enjoyable and fruitful conference. And hopefully, finally, this is my expectation to our rector, to Profesor Dr. Harun Joko Prayitno M.Hum.
Selamat pagi, makasih Pak Dekan.
Would like to open officially this international conference. So this conference hopefully will be opened by our rector. Thank you for you all. Nasru minallah wa batun qarik wa basril mu'minin. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Thank you, Profesor Dr. Anam Sutopo M.Hum, for the welcoming remark. The next agenda, the opening remark that will be delivered by the Rektor of Universitas Muhammadiyah Surakarta, Profesor Dr. Harun Joko Prayitno M.Hum, and later will open the conference officially. Professor Harun, time is yours.
Terima kasih. Thanks to Master of Ceremony ya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Alhamdulillahirabbil alamin. Wassalatu wassalamu ala Rasulillah wa ala alihi washabihi wa wala. Selamat pagi from Solo Sintra Java Indonesia. Good morning everybody from Solo Sintra Java Indonesia. Solo as the spirit of Java ya. Especially Solo, Surakarta, surround the honorable featured speakers. The first, thanks so much to Professor Mathew Seymour, Ph.D. from the University of Hong Kong. Good morning from Indonesia, Prof. And then the second, to Professor Dr. Engineering Shafi Samaun from King Abdul Aziz University. I think today several maybe twice or three times you as a invited speaker to UMS and then my colleague Mas Muhammad Lutfi Hidayat PhD 3 years ago take PhD from King Abdul Aziz and now he is as a lecturer at Universitas Muhammadiyah Surakarta at Education Faculty. Distinguished guests, ladies and gentlemen, all our participants. First and foremost, let us extend our praises and gratitude to Allah Subhanahu wa Ta'ala, the most beneficent and most merciful. May we also seek Allah blessing of Selamat and salam upon the Prophet Muhammad, peace and blessing be upon him. Good morning and welcome to the 4th International Conference on Biology Education, Natural Science and Technology, or INCOBEST. It is both an honor and a privilege to address you today in my capacity as the President of Universitas Muhammadiyah Surakarta. Now, Universitas Muhammadiyah Surakarta have 2 campuses, one main campus in Surakarta and the second campus in Denpasar, Bali. I extend my warmest greeting and sincere gratitude to all the distinguished guests, esteemed speakers, researchers, educators, and participants, of course, to my student from Biology Department education faculty who have joined us from across the globe to take part in this significant event. Today, we are honored to host distinguished invited speakers who have made outstanding contributions in their respective fields. This international conference under the theme Emerging Issues on Natural Science, Environment, and its Learning Innovation aim to explore recent scientific advancement in natural science and the environment, discuss their educational implications, and of course, share innovative pedagogical practices. Recently, Universitas Muhammadiyah Surakarta has been recognized as one of Indonesia's leading universities, as highlighted in the THE, the Impact Rank since 2024 until now. Additionally, UMS has achieved the distinction of being ranked as the number one and the best Islamic university in Indonesia. According to the TSA World University Rank WUR 2025 until now 2026. Furthermore, the university's research quality attained an exceptional score of 5.8.2, positioning it is the institution with the highest research quality in the nation. As the leading private university in Indonesia, UMS remains steadfast in its commitment to advancing technology for the benefit of humanity. So, in the faculty level, we have collaboration both in national and international level. We have in Faculty of Education 11, maybe more, 11 undergraduate programs, 5 or 6 master degree program, and 3 doctoral program of 90% which have excellent accreditation from BANPT Indonesia, from IAB, from LAM, and so on. In the last 10 years ago, we have massively arranged students such as delegating students for internship programs and students exchange to neighboring countries. Sub-exchange such as visiting professor, joint research, joint public services, joint publication, joint editorial team in several journals published by UMS have also been implemented with both national and international institutions. This event underscores our unwavering commitment to achieving the status of a world-class university by 2029. Through this initiative, we aspire to strengthen the relationship and cooperation between UMS and its partner institutions, fostering mutual benefit and engaging the impact of research and publication for all involved. I am pleased to note the remarkable response to this conference, and which has attracted almost 200 participants. Good, yeah? 200 participants and presenters. Yeah? The submitted article reflects cutting-edge research advanced in biology, natural science, technology, and of course, education commonly has significantly reshape our understanding of life, ecosystem, and environmental sustainability. It is highly encouraging to witness such robust engagement from researchers and educators across the globe. Developing a detail of the distribution of INCOBEST 2025 on 2026 participant. Three speakers. So today I think it's in the important conference. Yeah. In conclusion, I would like to convey my deepest gratitude to the organizing committee who Prodi, the scientific committee, and all those who have contributed tirelessly to the success of this event. Your dedication, your motivation, and effort have created this increasing platform for collaboration and intellectual exchange. Once more again, I extend my warmest welcome to all participants, speakers, and distinguished guests. Thank you so much. I wish you a productive and memorable conference experience. And we'll be open this conference together by basmalah together. Bismillahirrahmanirrahim.
Bismillahirrahmanirrahim.
Ya, semoga Allah SWT meridai dan merahmati kita semuanya. Enjoy in the conference. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof.
Thank you, Professor Harun, for officially open the conference. Ladies and gents, soon we'll be join the main event, but before that, let's give some preparation time to our keynote speakers and enjoy this video presented by Biology Education Department.
Ya, merah. Padahal aku udah berharap banget di jalur ini.
Kamu ngapain sendiri di sini?
Oh, lagi kepikiran hasil SNBPku kemarin, Kak. Jadi aku udah kepikiran.
Emang masih ada jalan lain. Hari ini itu bukan titik terakhir dari semuanya. Kamu masih bisa gapai cita-cita kamu. Masih ada kampus yang keren-keren di sana yang menantikan kamu.
Tapi aku berharap banget sama jalur SNBP kemarin. Aku ngerasa kayak aku bisa banggain kedua orang tua aku lewat jalur itu. Sekarang aku jadi ngerasa jauh banget sama cita-cita aku, Kak.
Tenang, masih ada jalan lain. Yuk kita explore bareng. Yuk. Udah sampai kita.
Jadi gimana, Kak?
Jadi kita sekarang ada di jantungnya pendidikan biologi UMS. Masuk aja yuk, kita kenalan sama yang punya rumah.
Boleh?
Program Studi Pendidikan Biologi FKIP UMS berdiri sejak 1979 dan saat ini terakreditasi unggul. Visi keilmuan kami adalah mengembangkan pendidikan dan pembelajaran biologi yang islami berlandaskan prinsip-prinsip pembelajaran yang inovatif dan adaptif berbasis kecakapan keilmuan biologi dan bioentrepreneurship. Visi keilmuan kami ini tidak hanya sekedar menjadi arah, tetapi benar-benar kami integrasikan di dalam kegiatan pembelajaran, penelitian, maupun pengabdian kepada masyarakat. Lulusan kami dipersiapkan untuk memiliki kompetensi yang luas, yaitu pertama sebagai pendidik di bidang biologi, yang kedua akademisi dan peneliti biologi, ketiga pengelola laboratorium biologi, dan keempat bio-entrepreneur yang inovatif dan mandiri. Kurikulum yang kami terapkan berbasis OB dan kurikulum ini memberikan kesempatan kepada mahasiswa untuk mengembangkan potensinya baik dari sisi keilmuan, keterampilan praktis maupun soft skill yang semua itu dibutuhkan di dunia kerja dan kehidupan bermasyarakat. Proses pembelajaran di biologi dilaksanakan secara terintegrasi antara teori dan praktikum, baik yang dilakukan di ruang lab maupun di lapangan seperti kegiatan PKL dan KKL. Semua kegiatan itu didampingi oleh dosen-dosen yang handal dan kompeten di bidangnya masing-masing. Dari sisi fasilitas, kami didukung dengan sarana dan prasarana yang lengkap dan modern, seperti ruang kelas dengan standar classroom dan laboratorium yang unggul, seperti laboratorium riset, laboratorium kultur jaringan, laboratorium budidaya jamur, pangan gizi, mikrobiologi industri, greenhouse, dan juga kebun biologi. Mahasiswa juga memiliki banyak kesempatan untuk berkembang melalui riset bersama dosen, kegiatan pengabdian pada masyarakat, program student mobility, KKN DIK, baik yang dilaksanakan di Jawa, luar Jawa, maupun luar negeri, hingga berbagai ajang pengembangan diri seperti PKM, Mawapres, dan kompetisi di tingkat nasional maupun tingkat internasional. Selain itu, UMS juga menyediakan berbagai macam beasiswa seperti beasiswa Kiai Haji Ahmad Dahlan, beasiswa Lunas Unggul Muhammadiyah, beasiswa Tahfidz Al-Quran, beasiswa unggulan, beasiswa prestasi, beasiswa internasional, hingga beasiswa KIP. Dengan berbagai keunggulan tersebut, kami mengundang putra-putri terbaik untuk bergabung bersama kami. Mari tumbuh, belajar, dan berkontribusi bersama Prodi Pendidikan Biologi FKIP UMS. Biologi FKIP UMS utama.
Makin yakin Kak daftar sekarang. Yuk aku temenin daftar. Ayo, yuk.
Science and Technology, INCOBEST 2026. My name is Yasir Siddiq, and it is a great honor for me to serve as your moderator for this session. INCOBEST 2026 is held under the theme of Emerging Issues on Natural Sciences, Environment, and its Learning Innovation. This term reminds us that advances in natural science, environmental research, technology, and education must be connected to address the complex challenges of our changing world. Today, we have three esteemed speakers who will share their insights with us, and each speaker will have at maximum 45 minutes for their presentation, then followed by 10 minutes Q&A session at the end. First, it is my privilege to introduce our first speaker, Mr. Muhammad Lutfi Hidayat, Ph.D. from Universitas Muhammadiyah Surakarta. Okay, first we will share the short biography in our screen first. Committee, please. Okay. Dr. Muhammad Lutfi Hidayat is a prominent lecturer at the Biology Education Department at Universitas Muhammadiyah Surakarta. And currently he serves as the head of Learning Resources Development and Scientific Publication Unit at the Faculty of Education. He holds a bachelor's degree and master's degree at Biology Education from Universitas Negeri Yogyakarta, Indonesia, as well as a Ph.D. in Computer Information System from King Abdul Aziz University, Saudi Arabia. His research focuses on digital competence among pre-service teachers, generative AI in education, digital citizenship, and the development of technology-based learning media. And before we begin, I kindly remind all participants to keep their microphones muted during the presentation, and questions might be submitted through the chat box and will be addressed during the panel discussion session. Well, are you ready, Dr. Muhammad Lutfi?
Yes, inshallah.
Yeah. Without further ado, let's begin our first presentation. It is now to great pleasure to invite Dr. Muhammad Lutfi Hidayat to deliver his presentation with the title of From Global Bibliometric Insight to Local Action, Advancing Citizen Science and AI for Biodiversity Education and Conservation in Indonesia. Please give a round of applause to Dr. Lutfi. And to Dr. Lutfi, the virtual floor is yours for about 45 minutes.
All right. Thank you, Dr. Yasir. Thank you for having me here. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
All praises to Allah. Alhamdulillah. Allahumma salli ala Muhammad wa ala alihi wa sahbihi wa sallam. Sorry. Is my voice clear? I would like to share my presentation. Alhamdulillah. Good morning, distinguished guests, fellow researchers, educators, and friends. It is an honor to stand before you today at INCOBEST 2026 to share a journey that began with simple question: how we can leverage the power of technology and community participation to document and protect Indonesia's extraordinary biodiversity. And over the past several years, my team and I have been working on a roadmap that connects global research insights with local action Today, I will take you through 4 parts of this journey. First, the background and the research roadmap. This is why Indonesia needs citizen science and AI for biodiversity. Second, the concept of citizen science itself, its global development, potential and persistent gaps. Third, our dissemination effort through peluru taloka and community engagement. And finally, our vision for the future, a deep learning-based decision support system for Indonesian medicinal plants. Let us begin. Part 1, let me start with a fundamental question. Why Indonesia? Indonesia is a mega biodiversity powerhouse. We are home to over 30,000 plant species. That's more than 10% of the world's known plant species. Of this, approximately 9,600 species are known to have economic and medicinal value. Around 40% of our plant species are endemic. They exist nowhere else on Earth. This is an extraordinary heritage, but here is the challenge. Much of the biodiversity remains poorly documented, and many species are threatened by habitat loss and over-harvesting. So we have a treasure chest of biodiversity, but we are losing it before we even fully know what we have. And now here's where the opportunity lies. The digital generation is an opportunity. Indonesia has one of the youngest and most digitally connected population in the world. According to Association of Internet Indonesian Network, 78% of Indonesian teenagers aged 12 to 18 own a smartphone. They spend an average of 7 to 9 hours per day on their devices. Currently, most of this time is spent on entertainment and social media. But imagine if we could channel even a fraction of this energy toward documenting our biodiversity. This is not just wishful thinking. Similar approaches have worked globally. eBird, for example, in the US, iNaturalist worldwide, and even in Indonesia, initiatives like Burungnesia have shown that young people are eager to participate in citizen science when given the right tools and incentive. The digital generation is not the problem, it is the solution waiting to be mobilized. Now, let me address the technological gap. There are excellent global AI identification tools available. PlantNet, iNaturalist, Google Lens, etc. For example, PlantNet covers over 84,000 species. iNaturalist has more than 500,000 species in its database. These are remarkable achievements. But here is the problem. These platforms are dominated by flora from Europe and North America. They were trained on datasets that are heavily biased toward temperate regions. When you try to identify an Indonesian medicinal plant, say a species like curcuma or rare orchid, the accuracy drops significantly. There is currently no application specifically designed for Indonesian medicinal plants, with a locally curated database and models trained on local species. And the databases that do exist are not integrated with AI-powered identification tools. And this roadmap has guided our work over the past several years. In 2024, I will make it short, we began with a global bibliometric study, mapping the landscape of citizen science research worldwide to understand where the gaps and opportunities are. 2025, we moved to from analysis to action, conducting teacher training and dissemination activities in Solo Raya region. In 2026, this year, we are developing our deep learning model for medicinal plant identification. In 2027, we plan field testing and expert validation. And by 2028, we aim to launch a mobile application and integrate it into school curricula. This is not a solo effort. It requires collaboration across universities, schools, communities, and policymakers. And I am grateful for the partnership we have built along the way. Part 2. Yeah, before we go further, let me clarify what we mean by citizen science. Citizen science is defined as the active involvement of the general public in authentic scientific research processes. This includes data collection, observations, analysis, and even research design. The core principle is democratizing science, making it accessible, participatory, and impactful. Citizen science is not new. For example, the Christmas bird count in United States began in 1900, year 1900, 1100. But what has changed in recent years is the scale. With smartphones, internet connectivity, and AI-powered tools, citizen science has moved from a niche activity to a global movement. According to Bunny, 2009, citizen science has become a developing tool for expanding science knowledge and scientific literacy. And citizen science is not one size fits all. There is a spectrum of participation. At the most basic level, we have the contributory model. This is where the citizens collect data following protocols designed by the scientists. Most large-scale platforms like eBird, iNaturalist, and PlantNet operate on this model. It is effective for massive data collection, but limited in citizen agency. At the intermediate level, we have the collaborative model. Here, citizens also help refine protocols, analyze data, and interpret results. This is common in community-based water quality monitoring programs. At the highest level, we have the co-created model. Citizens and scientists work together from the very beginning, formulating research questions, designing methods, collecting and analyzing data, and disseminating findings. This model is most empowering, but also most resource-intensive. Our work with Peluru Taloka, I will explain it later, sits at the collaborative and the spectrum, with aspiration toward co-creation as we engage more deeply with communities. Let me give you a sense of the scale of citizen science today. eBird, a citizen science platform for bird observation, has accumulated over 1.5 billion bird sightings. iNaturalist has documented more than 100 million observations of over 500,000 species. And here's a staggering fact. Since 2010, more than 80% of the data aggregated in the Global Biodiversity Information Facility, or GBIF, the world's largest biodiversity database, has come from citizen science platform. Why is this important for biodiversity? Because citizen science provides massive spatial coverage that professional surveys cannot match. It enables continuous temporal monitoring, data collected year after year, decade after decade. It is cost-effective, and it democratizes the data collection, allowing anyone with smartphone to contribute to global science. I will highlight this. It's allowing anyone with a smartphone to contribute to global science. Okay, next, our bibliometric analysis of 200 highly cited publications from 2000 to 2025 reveals some important patterns. First, there has been exponential growth in citizen science publications since 2010. This is not accidental. It coincides with the rise of smartphones, mobile apps, and platforms like eBird and iNaturalist. Second, the field was anchored by foundational papers published between 2009 and 2012, works by Boni et al., Silvertown, and Sullivan that established the theoretical and methodological foundation of citizen science. And third, we identified 4 thematic clusters in the literature. First, bio. This is a category for ecology, conservation, and species distribution. Public, for participation, crowdsourcing, and engagement. Data, methodology, data quality and machine learning, geo, spatial analysis, GIS, and remote sensing. This shows that citizen science is not just about data collection. It is about participation, technology, and spatial analysis working together. Now, let me show you a troubling finding. Our analysis shows that citizen science research is heavily concentrated in the global north. I highlight global north. The United States alone accounts for nearly 60% of all highly cited publications. The UK contributes another 9%, Germany about 5%, China about 4%, and the global south, including Indonesia, Brazil, India, and African nation, which has mega biodiversity, accounts for less than 2% of publication. Can you imagine that? This is a profound imbalance. The regions with the highest biodiversity, tropical regions like Indonesia, are the least represented in citizen science research. The knowledge we have about citizen science is largely built on temperate ecosystem and high-income countries. It may not translate well to tropical contents. This is a gap we must address. There is also a taxonomic bias. Birds account for 54% of species-specific publication in citizen science. Mammals account for another 14%. Invertebrates, plants, and other taxa receive far less attention. Why does this matter? Because the species that are most ecologically important and most threatened are often the ones that are least charismatic and least monitored. Lichens, for example, which are excellent bioindicators of air pollution, like every research, are almost entirely absent from citizen science datasets. Fungi, soil invertebrates, and many tropical plant species are similarly neglected. This is not just an academic problem. Conservation priorities are shaped by available data. If our data are biased, our conservation priorities will be biased as well. Part 3, dissemination efforts. I will explain about Peluru Taloka and community engagement. Let's bridging global insight with local action. I will introduce Peluru Taloka. What is Peluru Taloka? Peluru Taloka is a platform for citizen science-based outdoor learning. It connects students, teachers, and communities with local biodiversity with a specific focus on Indonesian medicinal plants. The name has meaning. Peluru is an abbreviation, pembelajaran luar ruangan. Peluru, pembelajaran luar ruangan, or outdoor learning. And Taloka comes from tanaman lokal, or local plants. Together, they capture our vision of channeling community energy into documenting and preserving biodiversity knowledge. This is the user interface for Peluru Taloka. Our mission is threefold with this Peluru Taloka: to document Indonesian medicinal plants through participatory observation, to integrate citizen science into school curricula, to build a locally relevant biodiversity database that can support research and conservation. The platform is live at www.pelurutaloka.com, and we invite all of you to explore it. Let me walk through how Peluru Taloka works. The workflow begins with students or teachers engaged in outdoor learning or project-based learning activities. They go out into their schoolyard or community, find a medicinal plant, and photograph it. Then they identify the plant using AI tools like PlantNet or iNaturalist. They take the geolocation using GPS on their smartphones, and they write a description of the plant and its uses with assistance from generative AI, like ChatGPT and Gemini, etc. They upload everything to the Peluru Taloka platform. Then comes the quality control step. The submission is reviewed by an expert botanist. If it meets quality standards, it is published. If revisions are needed, the student or teacher revises and resubmits. This workflow ensures that every entry on Pluraloka is scientifically credible with still being accessible to non-expert. And now let me share what we have actually done with Pluraloka. In 2025, we conducted teacher training workshop in Solo Raya region. 48 teachers from 20 schools participated. The workshop focused on AI-assisted species identification using PlantNet, iNaturalist, and generative AI tools. The results were encouraging. 90% of teachers reported feeling confident using AI for teaching after the workshop. 70% said they were more confident about integrating citizen science into their classrooms. Teachers also reported that hands-on practice was the most valuable part of the training. They appreciated being able to go outside of the class, photograph plants, and identify them in real time. This was covered, alhamdulillah, thank you for UMS Media and Sieratmu. It is a local media from Muhammadiyah in Klaten region. And it was a significant step in building local capacity. We have some photographs here, the workshop, the outdoor learning, practical work outside, and another teacher workshop located in this campus. Alhamdulillah, for the teachers accept this workshop well. And this is the dissemination, teacher training in Solo Raya, the activities in several schools in Solo Raya region. Okay, let me continue. We also took the initiative to the community level. Through community service programs with university students, we planted medicinal plants in several villages in the Solo Raya region, including Pabelan and Kartasura, and also Klaten. We are projecting to plant 100 plus medicinal plants, raising community awareness about local biodiversity. These plantings are also serve as a data source for Pluraloka. Students and community members observe, photograph, and document these plants. The activity is integrated into student research projects and coursework in botany and ecology. It is a concrete example of how community engagement can generate both conservation outcomes and educational opportunities. Let me share some key lessons from our dissemination efforts. First, one-day workshop build awareness but not confidence. What a pity. We measured a gap between perception of AI potential, 4.808 out of 5, and readiness to implement, 3.67 out of 5. And people understand the value, but they are not yet confident to do it themselves. Sustained mentoring is essential. Second, a community of practice is critical. Our WhatsApp groups became spaces for sharing success, challenges, and best practices. Peer-to-peer learning is powerful, I guess. Third, engaging school leaders is the key to sustainability. Without policy support from school leaders and resource allocation from school leadership, innovation faith will not go anywhere. Top-down support and bottom-up engagement must work together. Fourth, local context matters. Apps or application designed for temperate region need adaptation for tropical biodiversity. Language, cultural relevance, and local ecological knowledge all matter. Part 4, the last part of my presentation. I used Gemini AI for this infographic. Now, let me share. This is our vision for the future. We are developing a deep learning model specifically for Indonesian medicinal plants. Why is this necessary? First, global apps like PlantNet and iNaturalist have low accuracy for Indonesian species because they were trained on temperate datasets. Second, local medicinal plant databases are not integrated with AI. Third, taxonomic expertise is rare and expensive. We cannot rely solely on experts to identify every plant. Our solution is to train a deep learning model. Deep learning is different with our Minister of Education program. This is part of AI, machine learning, deep learning. It's different with our Ministry of Education program. Yeah, our solution is to train a deep learning model using transfer learning with architectures like ResNet 50 or EfficientNet. This is algorithm's name in deep learning. We aim for at least 85% top 1 accuracy on 500 or more medicinal plant species local from Indonesia. The model will be deployed as a mobile application, making identification accessible to anyone with smartphone. And building a deep learning model requires a high quality dataset. Our target is 10,000 images of 500 medicinal plant species. We are collecting these images from multiple sources: field collection in Soloraya, collaboration with Indonesian Medicinal Plant Conservation Center, partnership with local universities, and citizen science contribution through Plurutaloka. The challenges are significant. Indonesia has thousands of islands, and we need geographical coverage across this vast archipelago. Plants look different in different seasons, so we need images from various times in the year, and every image must be expertly verified. We cannot afford mislabeled data. This is a collaborative, multi-institutional effort, and we welcome partners who want to contribute. This is the, flow chart or pipeline from citizen science observation to decision support. Yeah, okay, I will pass this because I already explained before. But for short, from this database, we can support multiple stakeholders. For researchers, can access distribution data for conservation and ecological studies. Industry can verify the authenticity of herbal materials and source sustainably. Communities can learn about the plants around them and contribute to conservation. This is just not an app. It is an ecosystem for biodiversity knowledge. And who benefits from this system? Scientists and botanists at the first list. And they get accurate distribution data for conservation planning. The herbal industry gets verified raw materials and sustainable sourcing information. Students and teachers get hands-on learning opportunities and improve science literacy. Local communities gain awareness of their biodiversity heritage and cultural preservation. Policymakers get data for monitoring the Sustainable Development Goals, particularly SDG number 3, health, SDG number 4, education, SDG number 15, life on land. And conservationists get early warning system for threatened species. This is a system designed for impact across multiple sectors. And, okay, I will pass it because I already mentioned before. And now, of course, this movement faced challenges. Let me be transparent about them. First, we have a limited data set for Indonesian species. The solution is collaboration across institutions and crowdsourcing through Plurutaloka. Second, internet connectivity is a challenge in rural areas. We are designing, hopefully, an offline mode with chatting and synchronization when the connectivity returns. Third, expert validation is a bottleneck. There are not enough botanists to verify every observation. Our solution is a hybrid approach. AI initial screening, then expert verification for a subset, and community voting for the rest. Fourth, sustainability. We are developing a non-profit model, integrating with school curricula, and building institutional partnership. Fifth, language and cultural barriers. We are localizing interfaces and ensuring culturally relevant content. These challenges are real, but they are not insurmountable. Okay, for conclusion, let me conclude with 4 key messages. First, Citizen science has enormous potential for documenting Indonesian biodiversity, but it remains hindered by geographic and taxonomic biases. The global north dominates and charismatic species receive disproportionate attention. Second, Plurutaloka provides a platform to bridge these gaps through an ecosystem of citizen science, AI, and education. Third, our dissemination to teachers, students, and communities has shown positive impact. Almost 70% implementation, but sustained mentoring is required. Fourth, our vision, is a deep learning-based decision support system. This is an application for Indonesian medicinal plants, integrating citizen science, AI, and local knowledge. This is a journey, not a destination, and we are committed to seeing it through. This is the last slide, a little bit provocative. Ignorance is extinction. We uncover the unknown. Indonesian biodiversity needs our eyes. Now I want to invite each of you to join this movement. For academics and researchers, let's collaborate on data collection and model development. Contribute to Plurutaloka's biodiversity database. Conduct research using citizen science data. For educators, let's integrate citizen science and AI into your curriculum. Take students outdoors for plant identification. Publish student work on Plurutaloka. For government and policymakers, support citizen science infrastructure aligned with SDG monitoring. Allocate resources for biodiversity education. For communities, plant medical plants in your neighborhood. Document plants you find. Share local knowledge. Citizen science is not just about data. It is about people, partnerships, and persistence. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Muhammad Lutfi Hidayat. And to all participants, please follow me to give round applause to Dr. Muhammad Lutfi Hidayat. Thank you very much, Doctor. And
Thank you.
You have already clearly explained regarding the citizen science, then digital technology database for biodiversity, and also ended up by Plurutaloka platform, and also the future development of Plurutaloka, including deep learning utilization, right? And then share it to the all stakeholders. Yeah. I believe that since many thumbs up when your presentation, during your presentation, Dr. Lutfi, I believe that will many questions from the participants, but please hold on and let's move to the second speaker of this conference. Thank you, Dr. Lutfi. Our second speaker is Professor Mathew Seymour, Ph.D. Good morning, Professor Mathew. He is already joined us today in this Zoom meeting.
Hi, yes.
Thank you, Professor.
One second, my apologies.
Okay. Take your time. First, we will share the short biography of Professor Mathew. And it is my pleasure to introduce Professor Mathew to all participants.
Yeah, anytime.
Yeah, thank you very much for your time, Professor. I believe it is very early morning in your time over there in Hong Kong.
Oh, all good. It's not too bad. I think we're an hour ahead of you guys.
Okay, thank you very much. First, Professor Mathew Seymour is a molecular and environmental DNA ecologist whose work focuses on using large biological datasets to understand how environmental conditions influence biological community. He established the eDNA and E-Ecology Laboratory at the University of Hong Kong, where he advances the use of environmental DNA for biodiversity assessment, ecological research, biomonitoring, and conservation. Ladies and gentlemen, my pleasure to invite Professor Mathew to deliver his presentation with the title of From DNA Barcode to eDNA Metabarcoding, Building the Foundation for Biodiversity Monitoring. Professor Seymour, the virtual floor is yours.
Thank you very much. And thank you very much for having me and inviting me to this. I will try to speak slowly. I tend to get a bit excited, but anybody can win me down if I get out of control.
Okay.
So I'm Matt. I'm a eDNA ecologist. And today's talk is going to be about mostly about eDNA and particularly eDNA metabarcoding. But building on that, the importance of barcode reference libraries and how those have formed the backbone for a lot of eDNA research that we've done. And I'll try to do this with some examples and some introductory material that kind of covers the broad basis of all this. And just to set the stage a little bit, in terms of why we want to do this kind of work is, and I believe it's important for all of you, relevant for all of you, is that we really much rely on understanding the world around us, and that entails understanding the biodiversity that we have and how that biodiversity is impacted by the natural world and human impact and global impact and whatnot. And this is very important because we need stable and reliable biodiversity because it builds the foundation of our ecosystem services that we rely on so much for, just economical, vital stability. And so understanding what impacts are influencing this important network of organismal, biological information is essential for us to survive as a human society. And there's many components of that. There's, basic environmental assessment monitoring. There's conservation programs to protect key species or indicator species. And there's also relations to managing invasive species that are moving in that we have potentially pose a threat, et cetera. And there are many core groups that we kind of use to assess this, and we kind of focus on these as part of my research. And I'll probably focus mostly on these because these are the most relevant groups for my research. It includes things like diatoms, a lot of different fish species, a lot of different insect species that are their larvae live in the water, and they directly are influenced by the environmental conditions. And so these are what we call biomonitoring groups. And we use these groups to try and assess the conditions of different environments that we are going to sample. And I'm going to focus largely on water, because that's my background. I also put two species in the bottom here. One is the great crested newt, which is an important species for early eDNA research. And the bottom right here is Asian carp, which is an important invasive species that's more important for eDNA research as well. And so biomonitoring is not a new subject. It's been ongoing for quite a while. People have linked species assemblages to environmental conditions for decades now. The issue probably is that we rely a lot on traditional sampling. A lot of different netting, electrofishing, kick net sampling, diatom scraping. These are great, and they were established, and they're well documented, and people have made them highly reliable in terms of actually assessing biological diversity in relation to environmental conditions. However, there are limitations to it in the sense that it's very hard to scale. These techniques, these traditional techniques, take a lot of expertise. You need experts usually on very specific groups in order to accurately assess these different species. You get an assemblage of 100 or so species in your kicknet. You need to have the specialists that are able to identify each individual species down to a sufficient identification, and to do it reliably and repeatedly. And an individual sample may take a day or even two, depending on how big it is. And so it's very difficult to scale if you're looking to do a wider approach to facilitate more robust and more accurate conservation programs or monitoring programs, especially in today's world where we're always constantly fed increasing amounts of data. And so to get around this a little bit is you can use something called environmental DNA, which is extracting DNA from environmental samples. And now these environmental samples can be anything, really. They can be anything relating to indirect sampling of an area or surface that the target or supposed target of species has interacted with. This is usually related to things like water, soil, air is becoming increasingly more popular. But this can also be things like feces, it can be surface swabs, it can be rainwater, it can be a lot of things. There's basically DNA fragments everywhere that you touch. And if you can devise a suitable sampling strategy, you can extract those fragments and figure out what species has been interacting with that area. This is great because it is a non-invasive and non-destructive way of sampling. You don't harm the natural environment typically. There's no need to. And then you don't endanger the species that you're looking to sample from. And this is very important for a lot of species nowadays that are heavily regulated. There's a lot of fish species that it's very, very difficult now to do electrofishing in parts of the world. And it can be damaging to actually sample a lot of invertebrates if you're worried about interrupting the stability or emergence time of some of these species. So it's a very win-win situation here. It's highly versatile. And once you get the DNA out, it becomes very, very standardized in terms of linking those fragments to the actual species. And this is just a slide showing some of the more common ways that eDNA has been sampled and the number of publications. So you can see the large number is related to water. Here, there's all water-based research. This is only 2017, it's a bit dated, but it still pretty much holds true. There's probably another section here for air. A lot of sediment research. So water and sediment are predominantly the major research areas that have focused on eDNA. But you also have a lot of surface-based research, a lot of trapping. This is like hair snares, indirect sampling from other species, and then air. This is probably going quite a bit. But you can basically sample anything to eDNA. There is some caveats here in terms of that doing eDNA research requires very stringent guidelines in order to ensure that you don't contaminate things. This includes in the field, in the lab doing the extraction, and also in the data analysis parts. I won't go into this, but there are some great literature pieces out there and a lot of examples out there about how to ensure that your samples are collected properly and extracted and analyzed properly to ensure that the data itself is robust and usable and reliable within your study and across other studies. But that's just something to keep in mind. And also to be mindful, I just want to put this out there, is that eDNA, it would be great for eDNA to be standardized across every living thing in every living situation, but also be mindful that eDNA is kind of a dynamic thing. It will behave differently depending on the source of its origination and also in the environment that it's collected from. So where some samples may last long or have a long time period, such as a deep sediment might actually be from many, many years ago, and other samples will degrade really quickly, like a really hot tropical stream will not, the eDNA in that stream will not represent a long-term signal. And this might be great because maybe you want something very recently, and so that sampling right there and what you get from that site will be reflective of the actual biodiversity at that given site. And just a quick overview of how this works, in case if anybody's curious. You take your sample. Here I'm just showing you a water sample. You will filter out the sample. You'll clump it down. You'll filter it. You'll concentrate it. And that gets placed into a standard extraction at that point. There's different ways to filter out. There's different ways to digest this, and so these will depend a little bit on the structure, but the overall process is highly standardized once you collect the material from your given environmental sample. Once you have your DNA, it's pretty simple in terms of the steps involved. It's not so simple sometimes troubleshooting this, because generally what you're dealing with is a very cosmopolitan, very diverse set of genetic material, and you usually only want a certain number of individuals, a certain number of small fraction of that's actually in there. This is an example of, like, if you want to go after a crayfish, you might have very few fragments of DNA in that sample, so you can't do very complicated genomics with this, but you can amplify it. You can use primers to try and pick out those fragments that you want. Use PCR to amplify them. You can even make them glow in the dark a little bit if you want to do single species detection. But generally, you want to somehow amplify your target species or target group of species using some kind of tailor-made primers that you will then use PCR to try and make it easier for you to see the genetic material that you want. And then you will either use a standard PCR, qPCR machine to go after individual species, or what we're going to talk mostly about is this metabarcoding aspect. And so this is probably a more common approach nowadays. qPCR is still out there, but it seems to be less and less of a targeted approach. It still has a lot of value. It's cheaper and easier to set up, but there's a lot more, much more data, much more information available in eDNA metabarcoding. And with sequencing costs nowadays much cheaper, it becomes a much more attractive option. It's more complicated though, because you don't know what you're getting in your samples. So you're relying on the data to be reliable. In order for the data to be reliable, you not only need to have good field and lab practicals, but you need to make sure that you have good reference data. So here you have your environmental sample, you've done your extraction, you've done your PCR, you amplify the fragments. You then do your sequencing to generate your data, massive amounts of data. But then you need to have a reference data in order to make the data analysis part actually doable. And this is what we'll talk about next. And so to build a reference database, we need to have usually some sort of link between the species that we actually that we have in the environment and their genetic sequence. We need to have a genetic reference that we can then cross-check our smaller sequences from our eDNA data set with. And we do this using DNA barcodes. And DNA barcode is ideally a unique pattern of DNA nucleotides that we can associate with individual species. These are generally, and the traditional one here is CO1. It's about a 600 to 800 base pair fragment that we can link to a lot of different animal species. There's different regions because unfortunately there's not one perfect barcode for everything on the planet. And so each kind of part of the kingdom has their own unique or preferable set of barcodes that we rely on. You can sequence the barcode region for most of these things. It's just that whether it's informative or whether you actually get species from it is kind of but hit and miss. But animals typically CO1, but you have other fragments that you can use, Site B, 12S, depending on different subgroups. Plants use usually RPCL and MAPK. This is a bit more complicated. Usually you use one primer to kind of narrow things down to one taxonomic group, and then you might use another primer to narrow that down further. And this is also something you can apply for other groups, including fish and protists and fungi. Fungi typically use the ITS region, and protists generally use 18S or 16S, depending on your group of interest. That's just in there. There's many different barcode regions, and depending on the group you're working with, you'll need to probably focus on a different one. But these are just the general barcode areas. And where do you find these? And the thing is, once you have these barcodes generated, you generally put them somewhere that the public can get access to them. And this is where we have things like NCBI, BOLD, SILVA. These are really, really essential databases that are accessible to the public to get barcode information and to get taxonomic information from, in order for them to use that for their project. So without this reference data, eDNA research is not going anywhere. And there's many curated databases. So NCBI is great, a lot of great information on there. It's a bit of a mess because it's not really designed to curate large data sets. So just be in mind when you use these things. But there are many existing databases that will curate data that is more accurate and more user-ready for use when linking species to their taxonomic identities. And so with that intro, I kind of would want to talk a little bit about some of the research that we have done in our lab, linking for us to better understand the biodiversity in Hong Kong, and then some of the research that we've done to kind of showcase how eDNA can be used for effective assessment of biodiversity and species and conservation management. And the first example I want to give is on Hong Kong mayflies. So Yan is a master's student who I was working with. She did an amazing job here in taking some mayfly larvae and generating a very comprehensive reference database that we then use to assess urbanization influence on biodiversity. And just so I have a little bit of a reference here, but mayflies essentially are a really interesting model group. They're a very diverse set of insects. The adults are very short-lived, but the larvae will live in rivers and freshwater systems for up to 2 years, depending on different species. They're a very old group, and so they have evolved to be very special, very specific to different environments, and different larvae require different environmental conditions in this regards. And so this evolution makes it very interesting in terms of linking them to their environmental locations. And because of this, they are a very important group of what we call EPT. They're the E in EPT, which is an important bioindicator group that we use for assessing water quality in freshwater, particularly in North America and in Europe. And I guess from more of a science societal group, they're a very, very important food source for fish. And so that a link to there for a bit more direct individual, I suppose. And why this matters for Hong Kong is because Hong Kong, if you've ever been there, it's a very interesting place. It's a very condensed city with some natural parts as well. It's about 70% of Hong Kong is actually not developed. It's very rugged. So a lot of the urbanization is highly concentrated. So there's a lot of people in a very small area, but there's also a lot of nature, which makes for a very interesting highly urbanized to natural gradient. And this allows us to look at the effects of urbanization on biodiversity, particularly in the freshwater system. And mayflies and so there's no biomonitoring, freshwater biomonitoring in Hong Kong. And that's partly due to the fact that they don't exactly know what they have in their freshwater aside from certain species that they focus on, which are usually dragonflies. But mayflies are essentially important because there's about 4,000 species globally, but there's only about a very, very small amount is actually understood in China in general. Although Hong Kong, we actually have a former Ph.D. student who was working on this back in the 80s, who initially characterized this using morphological assessments, but we don't have a good existing barcode database to allow for modern assessment of these things using traditional or using more recent molecular methods. So we wanted to check on the biodiversity of these species in Hong Kong, and then build a reference database that could be used into the future without having to worry about trying to dig out old records. So we went across 30 sites in Hong Kong, looking at the transition from nature to urbanized, taking kicknet samples. For the mayflies, we collected over 12,000 nymphs, larvae, which were then identified to 46 species, essentially. And then from there, we generated 144 barcodes. This effort over the course of about a year accounted for about 84% of the expected mayfly biodiversity, and we found 3 potentially new species in this process. And this just speaks to the power of using what we call integrated taxonomy, which is where you combine traditional morphological identification, which is highly required for barcode referencing, combined with linking it to CO1 barcode assistance. So oftentimes what would happen is we would get a bunch of specimens, we'd identify them as close as possible. However, we still have specimens that are very difficult to identify, so they use barcodes to kind of help us validate what these species were based on global accessible barcodes. And then also you can then assess whether something new is a new species or not by looking at its sequence in relation to other existing barcode sequences. So from that, we then were able to kind of cross-check whether something was new or whether it was potentially a new species or a different morph. And this figure is just showing the photographic variation of the species that we have across the different species, across our samples, and a little bit of summary of the main findings here. And very importantly from this is that we also not only generated the barcode reference library, but we also assessed the impact of this mayfly biodiversity across the urban to natural and gradient. And we found, a very clear representation of urbanization influencing biodiversity. Basically, heavily urbanized sites basically resulted in a complete loss of mayfly biodiversity, which correlates to poor water quality. And that's what connectivity also is showing directly. And then from this, we can also look at indicator species. So we not only produce a reference database to highlight the existing biodiversity that we can then take into the future, but also highlight that there are particular species that are linked particularly to specific environmental conditions. So we have a unique species here in Hong Kong that's linked to have heavily linked to highly oxygenated sites that we can use for future biomonitoring. Peri-urban sites, which are kind of in between sites, they're basically slightly abused systems. They're actually more diverse because they serve as an interface. We have a few more species there, and that kind of helps us indicate whether something is being degraded or not. So these are kind of species that kind of indicate like whether we should be aware of a changing environment and whether we should do something to try and mitigate those losses before it gets too late. Building on that, another reference that we helped make was this larger Piranha reference library, where we actually expanded the known biodiversity in Hong Kong from 17 species to 243. It's a major undertaking that we did for about 2 years. Han Wu was an amazing postdoc that basically led this project. And I'll just briefly go over this, but Chironomidae are non-biting midges. They're part of the Dipter order, and they have a very, they're super diverse group. They're very underutilized in terms of biomonitoring, largely because they're so small and they're very difficult to identify. And they're a perfect target for creating eDNA-based research because traditional assessments of these guys is so difficult and takes a lot of work, a lot of expertise. And so we wanted to, it's been a passion project of mine to develop this since I did my postdoc work. And just being able to come into Hong Kong and do this was amazing. Working in Hongwu was amazing in this regard as well. But here we took 5 ecologically important streams. We took 3 segments within each stream to get as much of the characterization of Hong Kong. These streams occurred across Hong Kong. One was in Long Island, Hong Kong Island, and 3 across the main section of Hong Kong. And from this barcoding work, we were able to identify or generate over 800 barcodes, 243 species, and potentially 261 bins, which are kind of, if you analyze barcodes, they actually suggest that there actually might be more species than what we suggested based on the genetic variation within that barcode area. But we were able to confirm 243. And then from this, again, we're able to not only generate the barcode references that expands it, 10, 20, 100-fold or 10-fold, but we're able to link individual specimens and individual species to actual environmental conditions. And then, so we have now we have this database in Hong Kong. The next step is to actually utilize it, right? And so this is another amazing bit where we not only developed the Chronomin biodiversity reference data, but then we're able to actually use it to actually facilitate a more robust suggestion for how we can facilitate biomonitoring in Hong Kong using modern molecular approaches. And that's what this paper is from Hongwu as well, where we designed an experiment to actually test whether we can use eDNA, an eDNA-like approach to facilitate a chironomid-based biomonitoring approach. And the benefit from this is that we utilize an existing system that was developed prior called CPET, which involves taking the exuvia or the pupil skins of chironomid that emerge during their emergence period. So usually what happens is these species will pupate, and the pupa will float on top of the water, and the adults will merge and then fly off to mate. These usually occur in mass, so these pupil skins kind of accumulate in large mats on the water surface that you can then scoop up. This provides, in a sense, a large amount of material, a lot of biological material that is basically being discarded by the target group that we then can basically take back, mash up, and extract the DNA from. Traditionally, you could actually assess the genus level or even family level or genus level assessment of these skins using morphological methods. Again, highly specialized. You need you need someone who actually knows what they're doing. But that was the traditional way of doing this method. So then we wanted to upgrade this and try and see if we could use it for a molecular purpose. We had to validate this though. But essentially what we did was we formulated this framework called meta-CPET, which utilizes meta-barcoding of pupil exuvia to facilitate an identification of these species groups. And so to test this, we wanted to use an experiment. We used 15 mock experiments and some, not only in the mock experiments, we also looked at some natural sites to see how this works. For the mock experiments, we used 26 different species that are showing a wide range of diversity across Hong Kong. This is just a scary plot showing the mock experiment design. And we did this in triplicate so that we could see the variation in our results, but these are the mock sets, and these are the species that we used in the mock experiments, and the major groups to show that we have a wide variety, diversity of existing species in this experiment. We also used 2 different primer sets to see if we needed a more species-specific primer, or if we could use a degenerate primer in terms of how we properly estimate or measure this diversity. And these are some results. We found really good congruence in terms of the mocks themselves were highly similar in terms of replication, which is great. So we showed that we could identify differences in our mock communities. We also showed that there was a high detection of species, be able to find 24 of the 26 species in this experiment. So we had high success rate using this method. And we were able to determine not only the species, but also the link to the environmental condition used from our natural sampling. but only with the species-specific primer. The degenerate primer didn't do very well. So primer choice does matter in this regards in terms of using this method. So while these methods are out there, making sure that you use the right molecular approach is also highly important. And here we say basically the importance here is that using species-specific primers or more species-targeted primers is highly important for getting accurate species or genus level information that can then be linked more directly to their environmental indicators. And this is just a summary slide showing the importance of primers. The species chironomid-specific primers did a much better job of actually identifying chironomids and didn't waste as much sequencing power. Generally, what happens when you use degenerate primers is that you waste a lot of the sequencing on trying to identify things that are not targeted in the sequencing data. And that's basically what it shows here. This is nothing too surprising, but it's basically a validation of already kind of existing or presumed existing knowledge in the literature. And then a bit more, I guess, more vertebrate example. I know that the last major barcode reference data set that we have generated was working with Bayan and Thomas. Bayan is a Ph.D. student in jackfruit, passionate individual, and a very good fish expert. I don't think there's anybody more passionate about fish than Bayan. But we did a really cool per scene fisheries survey work here, where we characterized per scene fisheries in Hong Kong, which again, there's no existing barcode database to facilitate regular monitoring of these species in Hong Kong. And so there's over 1,000 fish species in Hong Kong, but there's no existing barcode reference database. So we wanted to try and utilize this is basically kind of carrying on from some existing work that we had to do anyways. And so we took all of this fish species that we were working on for this other project for the government and generated barcode references to potentially help us, if not help others, to do more accurate eDNA research in the future. But per scene fisheries is a very important part of Hong Kong fisheries. And so this potentially is really big for kind of keeping track of a lot of really economically important fisheries species, a lot of pelagic fish, cephalopods, some crustaceans. And so this was occurring over about a course of a year. We did this for about 9 months, sampled twice every 9 months, and mostly occurring in the eastern waters here on this picture. And this is a picture of Thomas working with the fishermen. So we would go out with the fishermen. This is all occurring at night as well. We do nighttime fishing and collect all the fish specimens that the fishermen were collecting during their night fishing over the course of 9 months. And we used 3 different barcode sequences to generate barcode libraries across all these species. We used multiple ones for each species. We used CO1 and 12S for the fish. We used 16S for the cephalopods and crustacean. And the reason we focused mostly on the fish, and the reason we used 2 for the fish is that sometimes sometimes one barcode reference is not really accurate for identifying fish down to species level. So sometimes we'd use 2 in order to help us increase the resolution. It proved to be really important for generating this database. But overall, this is a picture of the overall database kind of structure. We did 562 specimens that we sequenced successfully, ended up being about 206 species generated for this reference barcode database, mostly fish, 185 fish, but also some cephalopods and some crustaceans. So quite a diverse data set. Represents probably maybe about a fifth of the fisheries, of the fish species in Hong Kong. So it's a good initial starting pad for this. And hopefully future work here will help identify a little bit more and hopefully make some. We've already kind of used this. I can't give results right now, but the Bayan's already done some pretty cool work using this to then link to eDNA work and finding some really cool temporal patterns in some important fisheries, especially with croakers. But I can show a little bit about, really quickly, about some work that we did prior. I did this work on eDNA Arctic char in UK a while back. And Arctic char is a very important economically and societal important fish. And Wales, where I was working, was the southern end of this range. There's a monitoring gap in terms of Arctic char because it's really difficult to use traditional methods to sample them in a lot of lakes. You can't get access to some of these lakes, and you can't actually physically go looking for these species, although they're a really high conservation concern. People want to know where they're at and how many are at. So I wanted to use eDNA to try and assess lakes that were not being regularly monitored and seeing if we could detect these fish in these lakes and whether we could assess whether translocation of species into certain lakes would be successful or not. And so this is more of a pilot study, I guess, but we chose 5 lakes. One that we knew had species, 2 that we knew that had species, 2 that we thought might be missing, and 1 that was a reintroduction. So pattern here is a well-documented lake with fish. We found, basically what we found char in this lake, and matches the expectations. Quellan is expected to have fish of a lower diversity or lower abundance, and that's basically what we found. We basically found these white dots are basically the sample sites where we found no species, and the green sites are where we found species. And here we did find less abundance, which matches the expectations for both these species. Krapnot is a lake where they were supposed to translocate species into the lake. However, it seemed to have failed. We never found any char in these lakes. And then these 2 lakes are lakes that are not regularly monitored, but suspected may have char, and we found char in both of these lakes. So here we can safely say that these 4 lakes were successful in detecting char. This was a failed translocated example. And then from that, we could also basically estimate the abundance of the char in each of these lakes to give managers a kind of a snapshot or expectation of this work. This is important because to do this traditionally, they would have to get permits, go out and fish, and collect these specimens from each of these lakes, which is very difficult to do. I can just go out to a lake, take samples from around the rim, and get a quantification estimate from my eDNA results. Do all of this within a matter of weeks, versus trying to take months to try and go out with teams to try and accurately sample these populations. Got a little ambitious with my expectations, so I'll just give one more example, and then I'll let you guys go. But so the last one I want to talk about is a more recent work we did in Sweden, where we wanted to look at the conservation effectiveness of this European green toad. This is a very important species because it's a species of concern in Europe. It's a bit threatened in Sweden, especially, which is where we did our study. And Sweden has many, many ponds, so it's very difficult to actually survey everything in Sweden. It's very diverse landscape in terms of water. And that's probably the problem with when you try to sample these small amphibians, especially when you're trying to sample their tadpoles. So this picture is kind of somewhat realistic in terms of the layout of a Swedish landscape in terms of a pond network. And green toads, like other small amphibians, takes a lot of work to do regular monitoring and assessment. This is true for things like great-crested newts. Wyoming toad is also notoriously difficult. But you're dealing with an environment that's highly marshy, highly wet, and very difficult to assess in terms of the biodiversity and the population standpoint. And also you risk, in terms of toads, you risk transporting fungi and other disease-related pathogens from one lake to another when you do this type of surveying. So we wanted to see if we could use eDNA to assess this particular species and also simultaneously try to assess the impacts of the environment and other biological indicators on this species. So we did this doing 2 stages. These toads are reared and bred and then their tadpoles are basically reintroduced to the lakes for the chance to try and translocate. So we wanted to see first if we could use eDNA to determine the translocation success of these species. And so we actually followed a organization called Norden's Ark that were releasing these tadpoles in some ponds. And so we took samples before they released the tadpoles and then samples after they released the tadpoles to see if we could indicate whether our particular methods was actually going to detect the species to begin with. And these are the 3 ponds that we sampled from. This is before introduction of the green-toed tadpoles, and this is after introduction of green-toed tadpoles. And this green line is the green European green toad, and these other lines are other species of amphibians. And so for each one, we found a positive indication of detection, whereas we also found detection of other species in there. So we just basically showed that our method works and it's highly accurate, which is nice. The next phase was actually sample across our intended range of species. And from here, we basically showed that we sampled from 37 sites, and the red site is basically where we have the translocation effort here. And the main thing is that we found a lot of birds, we found a lot of amphibians, we found a lot of fish, and from this we're able to basically assess where we wanted to basically find out where would be a good place for translocation to be done in the future. And from our biological assessment of the eDNA data, we basically linked that sites that had a lot of agricultural use, such as cattle, or competition for nesting space due to stickleback pressure, were probably not great for sites for these guys, whereas things that may have mitigated or indicated more less threatening sites that included things like mallard ducks and common crane might be more preferred sites, essentially. And so from this, we did an analysis to basically rank the sites based on their suitability for potential relocation, and basically suggest which sites would be more likely to be receptive of toads, which is basically what you're seeing here in this map. And that's basically what the syncatable insight is. By just using MCDA analysis, we can basically show which sites should be prioritized for potential translocation of the species in the future. This is all using eDNA. Cool. And with that, I will conclude. One, that the DNA reference biological libraries are the foundation for accurate eDNA species identification. We need these robust regional data sets for improving our taxonomic position, because the reason we're able to do this stuff in Sweden and UK is because they have existing databases that we could then directly link to species. And now that we have them in Hong Kong, we're able to do them more directly. We show that with M.Hum's second experimental setup. Without that database, there's no way we could do that experiment. So you need good reference databases to do eDNA research at a high level. But eDNA is extremely powerful. Once you have these databases in set, they are highly reliable in terms of detecting species, your target species, and showing very robust links between biodiversity and environmental conditions. And you can use them for very quick and effective means to assess the conservation status and to develop management plans to enable future management and biodiversity assessments. And with that, I'd like to thank you for your time. I'd like to thank my lab for their amazing work and for support over the years. Thank you.
Okay, thank you very much, Professor Seymour, and to all participants, please give great applause for Professor Mathew Seymour. And for Professor Mathew already explained powerful eDNA methods to monitoring many species, including biomarker species, and then fish, and also amphibian and its concurrents. I believe that many participants already have the question for you, Professor. But is it okay for you if we place the question session at the end of this session? Okay, thank you for your time, Professor. Ladies and gentlemen, now we continue to our third keynote speakers. It is a great pleasure to invite Professor Dr. Shafi'i Samaun. Good morning, Professor.
Good morning.
Good morning. Thank you for your time. Is it still early morning over there in Saudi Arabia?
Yeah, luckily I'm in Indonesia now.
Okay. So it is no problem for the distance, right? Okay. Let me introduce Professor Shafi'i Samaun first. Please, the committee show the short bio of Professor Shafi'i. Professor Shafi'i Samaun is a professor in process control in the Department of Chemical and Material Engineering at King Abdul Aziz University, Saudi Arabia. His expertise cover control theory, system engineering, process dynamics, artificial intelligence, reinforcement learning, energy system, robotic, healthcare engineering, and sustainability. His work has contributed to the development of intelligent control system with application in chemical processes, energy optimization, anesthesia regulation, and diabetes management. And today, Professor Shafi'i Samaun will deliver his presentation with the title of Riccati-based Edge Infinity Observer Controller Synthesis of Multiple Time Delayed System with Application to Diabetes mellitus. Please give a round of applause to Professor Shafi'i. And Professor Shafi'i, the virtual floor is yours for about 45 minutes.
Okay, thank you so much for allow me to do the presentation. First of all, before we start, let me share the screen first. Maybe you can check on, you can see my screen now. Yeah, as it is already introduced, so I will talk regarding the Riccati-based Edge Infinity Observer Controller Synthesis of multiple time delayed system with application to diabetes mellitus system, and I will explain how it is diabetes mellitus as a delay system. So I'm with Department of Chemical and Material Engineering, Faculty of Engineering from the King Abdul Aziz University in Kingdom of Saudi Arabia. I just finished this slide by yesterday. So still on July 3rd of 2026, okay? The overview of the talk will include on the introduction and then problem definition and then contribution definition and then some lemmas used in this contribution and controller synthesis. And I will jump to numerical application for diabetes system and conclude in the conclusion section. So as introduced before regarding who I am, Dr. Shafi'i Samaun, educated for a Ph.D. and Master in Automatic Control and System Engineering from the oldest university in Europe, University of Valladolid in Spain. And I have above 20 years experience in academic and also process modeling and control system and also machine learning, especially goes to reinforcement learning. And then, of course, as I come from machine learning and control theory, my expertise is on modeling and data analytics to the embedded system engineering and also advanced control design. And now I need some industrial linkage and also heat transfer laboratory. And also I'm a co-founder of Data Intelligent Analytics and Fiber Terra Network. Okay, so So let we begin our talk. Maybe I can finish the talk regarding 40 minutes given, maybe only for 30 minutes if it is possible. So let we define the delay system. Delay system, according to definition, is something happen now, but the response might come after the inquiry. So delay is a system often referred to in engineering and physics as a time delay system of transport delay. Okay, for example, in transport delay and constantly present in daily life. Today, we cannot avoid using this delay. For example, now we are using our network in communication to present this talk is also including the delay. Such delay in this system can be in the output or in the response, and an action doesn't happen instantly. It might cause something. Okay. For example, delay can happen in home automation and also appliances, and also in digital and communication network, and also in transportation, infrastructure, human biology, and nature. So I will explain today that delay in the human biology and nature, and also some other type of the device nowadays is contain the delay system. So in diabetes system, for example, we have we call ingestion. That means the amount of glucose we take, the amount of food we eat, and that is converted to be a glucose in our body system. And then for the glucose level is, the glucose as energy for all the system in our body. That energy can, activate many cell in our life. And from that glucose, the glucose level is maintained by the insulin. Okay, by the insulin. So that insulin mean a signal given by the glucose through the pancreas to produce insulin, and insulin will send the signal to the liver to regulate the glucose level. So high glucose can cause many problems, and low glucose also can cause many problems. So here, if we see the chart, what I prepare here, the glucose level is, send the signal to pancreas to produce insulin. So this production from pancreas have some delay. It's not immediately information can come up with the presence of glucose. Okay. Similarly, when the insulin level is high, the glucose also have to be maintained using the insulin. Okay, while the patient suffer with diabetes, that means that the level of insulin in the we call this in the blood is not sufficient, and then we need to introduce another insulin. We call insulin infusion. Okay, we call that insulin infusion. But sometimes the patient might experience the insulin production is in between low and high. So sometimes it can be low, can be high. So that's we call as the hybrid diabetes system. So what we mean hybrid, sometimes the patient can experience type 1 diabetes. Type 1 means we need totally insulin from the outside. And type 2 diabetes, sometimes the production of the insulin is not sufficient to treat the glucose in the blood system. Okay, and problem with time delay system, we mean time delay here is when we are dealing with the state coming to the system from the previous state, okay, from t minus 2. That t minus 2, this we call as the delay term. And in this delay term can introduce many characteristic root when we convert this linear time invariant system into the Laplace transform. Here, this term will give us asymptotically goes to 0, and that means we will have many roots of this system. This can cause the problem of the phase lag increase, and then the stability margin might decrease, and the oscillation appear is because of this delay, and the system might eventually become unstable. So this delay can cause instability in the system. When the system is unstable, it is mean that it is very difficult for us to control. This in diabetes system, when the delay is increased or the delay is high, the moment we are introducing the insulin is the next moment it can cause the insulin injection is very high and can cause hypoglycemic. When the patient experience with hypoglycemic, then he might lose consciousness. It means that he might get conscious. So when he might get conscious, he cannot do any activity. Okay, that's the problem. And if later on the controller corrected the insulin level, injection to the blood, then due to this delay, it might cause hyperglycemic. When it is hyperglycemic, can cause so many problems on the vital organ in the diabetes system might be disturbed. Okay. Performance deterioration also in the in the delay system. If even the stability is preserved, the delay often degrades control performance because the longer settling times, and then larger offshoots might come, and then the increased oscillation, and then reduce disturbance rejection, and slower tracking or reference signal. This happen in the time delay system. That's the problem in time delay system. Then how we must design the controller to include the delay itself in the controller system, and also reduce phase margin. Okay, delay contribute pure phase delay without affecting gain, meaning that the delay can cause the phase to be larger, or then the affecting the gain without affecting the gain. The gain is remain the same while the phase is changing. This can cause smaller phase margin from the smaller gain margin, then increase sensitivity to disturbance and higher likelihood of oscillation. Instability might happen in the time delay system. For delay system, we have also we call infinite dimensional as in nature. So the solution of these differential delay equation is only depend on the entire history. So depend on the entire history of this we call t minus theta until the t. So this we call this higher the entire history. Okay. Then therefore the system state become a function rather than a finite dimensional factor. Usually when we solve the ordinary differential equation, We need the initial condition. That initial condition is only 1 point, while in the time delay system, the initial condition we need, and we need the history of the previous condition. Usually we use as a function. So this creates several difficulty, more complicated mathematical analysis, and also infinite eigenvalue, and complex numerical computation, and higher computational costs that we need to do in numerical method. We need to do some computational that need cost, take time for the calculation. Okay, most controller design approach neglect the existence of delay, meaning we avoid the delay because delay here is quite difficult to include in the controller design. So therefore here we need to include the delay. So the delays exist, and this controller often requires significant modification when the delay is appear in the system. I give for example, when you tune the PID control from PID mean proportional integral derivative control. So the tuning point, when that tuning point of the system, and when the system has the observe the delay, that we have to retune the PID controller to modify somehow for the delay to happen, or to minimize the effect of this delay. Okay, and then another problem with uncertain and time varying delay. Time varying delay, most of the system what we are talking about, we don't know exactly how the delay behave. And my varying from time to time, the delay might vary from one sample time to the other sample time. So, for example, like in internet communication, like we are doing today, it is might cause some delay, and that delay from sampling time to the sampling time is not constant. It's always varied. And then for wireless sensor network, similarly happen. For cloud computing is also, and for remote robotic surgery is also have some delay in time varying, and also for working control system. Okay, for these many happen of time varying delay. So I will go with the diabetes later on to explain about time varying delay. Well, we use the state feedback control. We need somehow to observe another state if we cannot measure the state. Usually, we use a Leuenberger observer or the Kalman filter observer. But this observer in state delay is quite difficult to apply directly of the Leuenberger and Kalman filter. So the measured delay produce outdated information, meaning that the measurement right now can come from the measurement before, which can result in accurate state estimation and also slow conversion and degraded observed performance. This issue is critical in, for example, aerospace system, in autonomous vehicle, and in another industrial automation. For example, if I go with aerospace for, let's say right now we know that kamikaze drone and also intercept of the drone, that's we have to involve the delay system to be accurate information where the object is positioned. And also in, for example, in industrial automation, like in chemical engineering, for example, when we deal with, ethylene oxide reactor, for ethyl oxide reactor, we have to deal with precise controller. Otherwise, when the reaction is run away, then the explosion in chemical reaction might happen because this ethyl is reactive with the oxygen to have the ethylene in the petrochemical industry. This can cause very high difference in the exothermic reaction. So we must maintain the precise control for this kind of the problem. And also in communication constraint when we deal with networked control system or in communication system, such model control system often rely on communication network, the network control system, smart grid, multi-agent system, Internet of Things, industrial Ethernet, for example. And this communication might cause the transmission delay, packet loss, and then we have also jitter, sampling delay, and also the synchronization error. This factor collective reduce system reliability and control performance. And in time delay system, this one we must put into account for the consideration how the communication will happen, and we have to put that information. So another problem in time delay system is the computational challenge. Simulation of delayed system require storing historical state trajectory, what I mentioned to you before. So this one will be as a function or as the data will do required. And then this can cause the numerical solver must continuously interpolate previous state where the previous state is stored. Okay, and another problem with the delay system is modeling uncertainty. In practice, these delays are often difficult to measure accurately because it's unknown exactly. So it might include from the actuator dead time, from the sensor latency, and also from the transport phenomena, and also from the computational time, and also from the communication latency. This might cause the uncertainty high in time delay system. So this what we want to talk about the definition on my contribution in this article or in this talk. So let's say I call sim M, or meaning that the symmetric matrix of M is M plus M transpose. That's what I mean, symmetric M. And then I have the second definition I call Rm, which is the row matrix of M from M matrix 1 until M matrix N, meaning I have N matrix there. And then also for the diagonal matrix Mi, this is I will go from M1 until Mn, so to be the diagonal matrices. So the M itself, M1 and Mn is the matrix itself. Okay. And then we use this lemma, okay, Sur complement lemma. For example, we have the matrix M1 equal to M1 transpose, meaning it is we have the symmetric matrix, okay, and then this M2 has to be positive and also symmetric, and M3 having the inequality. So like this, we have M1 subtract by M3 transpose multiplied by M2 inverse and M3, and should be less than 0. This is satisfy if and only if this matrices is for the first on the left hand side is less than 0, and also for the right hand side is also less than 0. So the position of M1 and M2 is replacing in these matrices. Okay, and the proof of this matrix I didn't prove because this is very well known in control community for the Sur complement. So you can check any text regarding the Sur complement. Okay, and I have this lemma, which is the lemma when we have the matrix M again. This is positive with the size is M by M, is symmetric matrix, and we have matrix Z is Q by Q exists such that the Cm equal to Z, okay, ZC. So I just to replace the position of CM, C multiplied by M, and equal to Z multiplied by C. This might happen only if and only if the C, okay, that C has the size Q by M, for Q is less than M, having singular value decomposition. Oh, this is SVD, not SCD. Okay, sorry, this SVD. Singular value decomposition of the C is to be like this. Okay, we have U, C, and V, and the M itself have this kind of the matrix, this kind of the matrix, M multiple equal to V multiplied by M1 and M2 and with V transpose. And where M1 and M2 has the size Q by Q and M subtract by Q. So this to compensate, the idea is to replace the position of this one from C multiplied by M to be Z multiplied by C. Okay, how I use this one, I will show you later on in the theorem what I developed. So this is the proof regarding this lemma. We have C multiplied by M, and then we replace this M after we replace this C of the singular value decomposition. Similar also for this C, singular value decomposition. And then from there, we can see that the M itself will be this one. And this is telling us that this is happen when UCM equals CUC, then for M0 equal to 0. So this is how the proof is look like. Then we can apply the idea of CM equal to CC. Okay, so we do place the position. Now I come back to the idea for controller synthesis. A multiple time delayed linear system. What I mean time delayed linear system, we have tier 2i here, and this is multiple from I equal to 0 until n, and we have B and U. This is the input, internal input with the weight of B1, and this is the disturbance and with the weight of B2. Okay, and this is the output measurement. We have C multiplied by x of t, and this is the initial condition with the history of this function, with this kind of the history, which is we take the maximum delay function until the time 0. Okay, and that's all the size of the matrices is given here, which is A1 is M by M, and B1 M by P, and B2 M by Q, and C also R by M, constant matrices, and set, and then pi here is at the initial condition function. Okay, and we need to do the observer. In this case, I let the extended Leonberger observer, we call L, to be applied to the system having no disturbance. I try to minimize disturbance first to take out the disturbance. And the equation before and the equation 5 before, this equation now belong to be this type of the equation where the disturbance is replaced and then the Leonberger observer is applied. By considering that, now we take the U of t as the state feedback. We have K multiplied by head of X, where the K is the control again. That we will design later on what is K. What the K matrices is suitable for the kind of this problem. And we let the error of X to the X estimate or head of H according to observer is given by the error. This is the error at time t. Then the error dynamic of these two matrices, matrix system 5 and system 6, this will look like this. Okay, the error dynamic, now we have the error dynamics. This will be our matrix to weight to the error at the immediate time, and plus the weight of the error at the previous time, plus now I add back this type of disturbance where let to get between x and error, x coming from equation number 5 and the error coming from equation numbers 8, we combine it together, we join it together. Thus, the system in 5 and error in 8 can be presented to be this kind of the equation. This kind of the equation, which is now is simply, we have z dynamic equal to I equal to 0 until n, and we multiply by lambda I z t minus theta I plus b multiplied by omega wt, okay, by wt, and the output will be c multiplied by z. Okay, now this lambda I and this b is the augmented matrix. What we have this like this, we have lambda 0 and lambda 1 and so on. We can go for 1 until n following this lambda. Lambda 0 itself is A0 plus B1K, subtract B1K, and A0 subtract by LC. So what we learn from this is the, the closed loop behavior. For the first row is telling us the closed loop behavior. For the second row is telling us this is the observer to know where the unmeasured state. Okay, while B is the content B2 and B2, while C is content C and 0. And from there, we can take the Laplace transform to the system number 10 from the output epsilon t to disturbance function Wt is to have this one. To have this t, or we call this also t transfer function, from this output to the disturbance rejection. Okay, we have this kind of the problem, and we have to know what will be this inverse problem. And to solve the inverse problem is quite difficult. Then from this we try to get to stabilize of the system, closed loop system in system number 5. That's guarantee the H infinity. So the H infinity here we define to be the norm of infinity of this closed loop transfer function, of this disturbance transfer function, that such that this should be less than gamma, and this gamma should be strictly positive. And what we have to do later on is we try to minimize this gamma, and that minimization guarantee to be positive, and this will guarantee that the system number 5 will be stable. So this then a system presented in equation number 10, what we have here is we can represent this one in that fulfill H infinity performance, which is less than what we call transfer function, disturbance transfer function, in term of H infinity is less than gamma, and the gamma is positive for all tau is to be positive if the following Riccati equation is 4. So this is what we call Riccati equation. Okay, this Riccati equation, the first part. Now I use this sim p multiplied by lambda 0, meaning that here is p multiplied by lambda 0 plus lambda 0 transpose multiplied by p transpose, and the sum of I until m of these qi plus pi p multiplied by lambda I and qi inverse and lambda ip, and also for this type of the output function, and this for the input of the system. And this should be less than 0 or all of this. Okay, and then this one I didn't prove in this talk because it's quite long. So you can refer to my latest publication on how to prove this equation 13 and satisfy the H infinity from the Riccati equation. So then from that one, we try to develop this theorem. Okay, a system number 5, which controller law at equation 7, presented in equation 10, is asymptotically stable and satisfy H infinity norm bound. This we call H infinity norm bound less than gamma for the given gamma positive, and 2 is also, the delay function is also positive. If there exists positive definite matrices such as Y, and then Qi for I equal to 1 until M. There is 2 type of Q here. Okay, be careful on, you check this now. This Q, 1 Q with head, with tail, and 1 Q with no tail. So, and the matrices X and R such that the following linear matrix inequality is whole. So that this matrix pi, I call this matrix pi, should be less than 0 according to the Lyapunov sense. Now you see that I put here sim is already defined before, and also I put here also sim, I already defined before, and I put R here is already defined before, and I put this diac, I already defined before. Okay, and then you follow by this kind of the matrix. We need to do to solve these matrices to find the unknown X, Y, R, Q, and X, Y, R, Q, and Y, yes, Q and R. Okay, so we have to solve from that. So let's check the proof. Okay, according to applying the Sur complement, this is already defined in lemma 4 to the lemma 6 before. Okay, lemma 4 and to the lemma 6 before is already defined for this proof, and it has this kind of the problem. Okay, kind of this problem. Now, what do we see for this problem? How we can solve this problem? So this problem is we need to do the accumulation. For example, when we have this still in augmented matrix, okay, the omega lambda 0 and lambda I still augmented matrix, then we need to break up to be or we make it bigger to have this kind of the matrix, because now our objective is we know A0, we know B1, okay, we know this B1, we know AI also, we know this AI, but the rest like P, we don't know P, we don't know Q, and also we don't know, P and Q here, and K, we don't know K. So from this, what we have to do, because this we can cause, bilinear of P and K. When we multiple, P and K is unknown, so what we have to do, from these matrices, we need to move multiple both sides, on the left side and on the right side, by, we call these matrices, okay, the P inverse I. We multiply, because this 5 by 5 matrix, we multiply by P inverse I. This is the diagonal, okay, the diagonal I mentioned here. This is should multiply by the diagonal of this matrix, okay, and then we take Y equal to the inverse P inverse, and we take this, the q till with y multiplied by qy to have this kind of the equation now. So now on this hand side, this one k multiplied by y, okay, still b linear. This is bilinear, and also k multiplied by y here is bilinear, and then here we have LCY, and the rest no problem. Here is the problem. So LCY, L we don't know, and Y we don't know. C is in the middle. C in the middle. That's why we apply the lemma before, such that we can have like this. So LCY equal to ZC equal to RC. So L multiplied by Z we assume to be R. Okay. So now the C here is already removed to the right, while the Y here disappear because now will be Z. So that for the Z itself, now will be this kind of the solution. The same thing for K will be the solution of X multiplied by Y inverse. Okay, by this to calculate what will be K and Y. So from that we can use the corollary how to minimize the omega, where we want omega to be positive in the feasible solution by considering the following minimization problem. So we try to minimize gamma such that the gamma is to be positive, with subject to Y to be positive, Qi to be positive, both Qi to be positive, and also for pi to be negative, and gamma to be positive. That's what we want to go. We want to know. So we can solve to get what is K, L of the problem that we want to do. So I come back to diabetes system. This I took for a simple model we call the glucose dynamics of the first equation. This telling us the glucose dynamic. We have M in, this is the meal intakes. This meal intake in terms of glucose, okay, not in terms of meal itself, but we have to convert to the glucose level of the meal intakes. And then we have the function one, which is the insulin, the presence of glucose and of the presence of that insulin. The insulin can maintain the glucose by this kind of the information. This is also called the collagen conversion to the glucose by the presence of the insulin. And this is the function of the glucose, and this is such a function of physical activity, like sport, like exercise, whatever, physical activity. And for this case, we also have the insulin dynamics. We have U here is the insulin intakes or insulin injection, and beta here is the pancreatic function, with F5 here is the function, the insulin secretion due to the presence of the glucose, and this is the Maclaurin-Menten equation to see the elimination of the insulin in the blood system, in the blood plasma system. We call this also as the plasma system. And all the function F1, F2, and then F3, F4, F5 is the nonlinear function, which is give F1i, meaning I go with this F1, we give us this kind of the function. And F2 of the glucose function, which is glucose here, is this function. And F3 is the glucose function, this quite linear function. And F4 is the insulin function. We have this insulin. and F5 is the glucose function. We have this glucose function there. And from this equation, again, the delay function here, I take as the time varying delay, because we don't know what is the delay in our body system for the glucose intakes, so that the conversion times of TO2 of the glucagon or the glycogen to the glucose due to the present insulin is unknown. And also the insulin secretion time we call TO1 due to the presence of glucose is also unknown. So I assume this to behave as a sine wave, with this kind of di plus g and sine of omega t plus rho, which is the random number to present the uncertainty, for this talk. Okay, so that's for example, you can see photo 1 is behave like this, and photo 2 is behave. I behave like that. I assume to be behave like that. And for the pancreatics, also, for pancreatics, this function, for the beta pancreatic function, so the beta might goes to 0.2, around 2.2, something like that. Until there, I assume this the way of switching between type 1 and type 2 diabetes. What I mean type 1 and type 2 diabetes, when you are at less than 0.05, slowest number, or 0, meaning you are at type 1 diabetes. And when you are above on the beta is above this number, then you have the type 2 diabetes. So how type 1 and type 2 behavior, we can see later from this, the pancreatic function problem. And then we linearize the model, where the linearize of the model is looked like this. We linearize at G star and I star. So the A0 now is look like this equation, and A1 will look like this, okay, and A2 will look like this, where we have this zeta1 and zeta2, and the function of zeta1 and this zeta2 is given by this one. Okay, the prime here, the function prime is the derivative function, okay, of respective function. For example, function 2, derivative of the function at this g, and also the derivative of this the other function at that one. Okay, and b1 and b2 is given like this. Okay, that's given like this for b1 and b2 or itself, and c is c1 and 0. And the glucose intakes, I assume that the glucose intake is 3 times a day. 3 times a day mean every 8 hours the patient eats something, okay, and then this convert to be glucose. The food is composed to be glucose with amount of 1 milligram per deciliter, and this is behave like relay data function, so that when the time you eat, it is the glucose ingestion is high, and then degrade to be 0, and then after some time you eat again. So every 8 hours, the sequence is repeated with the same amount. This is what I try to do the simulation for this case. So by applying corollary A of the theorem 7, so we obtain the Y matrix like this, and the X matrix is also like this, and from that we can find the K is the the controller gain to be 0.0326 and minus 0.1305, and the L, the observer gain, to be this kind of the observer. Okay, we have this kind of the observer, and from here we try to do the simulation. What we find and we compare to PID controller. The PID controller cannot maintain when the patient goes to what we call diabetes type 1, while around for the diabetes type 2, the PID controller with the black color here is still can maintain, but we have very high oscillation for each meal is intake. For each food is we eat, the oscillation is happen. Well, for observer, the one we propose is the green light. The green light is still maintain very well the plasma glucose concentration or blood glucose level around 120, while oscillate during the meal intakes, but that oscillation is less than the PID controller. The best thing is if we can know the insulin level, what we call non-observer, meaning that the insulin level is feedback to the controller without observer, and this is work very well. But again, in real life, we don't have the sensor for the insulin. We have only the sensor for the glucose. So what we can see that here, the proposed system still work well inside the normal, bring the system to be the inside the normal blood glucose level, which is around 80 to 180. Usually around 80 milligram per deciliter until 180 milligram per deciliter is considered normal glucose level. When low glucose level, it can cause unconscious. That means that PID controller here can cause unconscious to the patient, and also hyperglycemic, and for the patient when it is experiencing type 1 diabetes. Why this happen? This because I try to linearize the system around type 2 diabetes. Type 2 diabetes. Maybe we can try to linearize in another point, so such that we can get better solution. Okay, that's part of conclude my talk on an optimal state feedback observer-based controller has been synthesized based on the proposed method of the extended Riccati equation for multiple time delayed linear system, and it is implemented numerically for hybrid diabetes system, hybrid main type 1 and type 2 diabetes system to maintain plasma glycemic level. Okay, the proposed controller with precise state observer has better performance compared to well-established PID controller. Again, like I mentioned, the weakness here because we don't have the measurement of the insulin. We have to do the observer for the insulin. We have to estimate the insulin level, so that we still have a problem, but anyhow it's accepted already. As insulin level is not able to measure, then the linear observer is applied. The state observer feedback control behave poor due to inaccurate insulin information, and therefore future exploration is to search a precise state observer method. I mentioned before in the early talk that when we deal with time delay system, the observer, Leonberger observer and Kalman filter observer directly cannot be used So I try to include the delay here, but still we need to improve what the observer is. So you can check I refer only to this one reference that I already published the work in the conference 2023 in Malacca, Malaysia. So you can read how I prove the Riccati equation from the defined problem before. You can check how the derivation of the Riccati equation is used. So I think I conclude the talk to the end of this talk. Maybe I spend some time.
Okay, it's okay.
Thank you.
Okay, thank you very much, Professor Shafi'i Mahmud. And to all participants, let's give great applause to Professor Shafi'i. Thank you very much, Professor. And your presentation has shown how advanced mathematical modeling, observer controller design, and intelligent control system that contribute to improving glucose monitoring in diabetes management. Ladies and gentlemen, and the speakers, now we will proceed to the panel discussion and question-answer session. Participants are welcome to submit their live or direct questions by raising hand or indirect question through our chat box. And we open for 3 questions first from the participant. Anyone will deliver the direct question or feedback to all 3 speakers. Okay. If during waiting the question from live or direct question, I would like to read some question in the chat box. Okay, the first is from, it is including feedback, right? Not only questions, but also feedback. And the first is from Faisal Ma'ruf for Dr. Lutfi. Is the curation has been supported by various fields of sciences related to plants data within the platform? So your time, Dr. Lutfi, you can answer it.
Okay, thank you, Dr. Yasir. Okay, thank you for the question, Faisal Ma'ruf. Okay, yes, for the curation, we have several experts from several disciplines. For example, for our botany team verifies the taxonomic accuracy of the plant data, and then we have the pedagogy team that ensures that the content is educationally appropriate for schools. And also we have the IT team, which developing AI-based tool, including deep learning, etc. However, we realize that biodiversity data require multidisciplinary expertise. Yeah? Therefore, from this conference, we are actively seeking collaborators with researchers in medicinal plants, pharmacology, ecology, taxonomy, and also computer science for further improve the quality of and coverage of the Peluru-Taloka database, like that. Thank you.
Okay, so the Peluru-Taloka already curated by many stakeholders, right, Dr.
Yes.
Lutfi?
Yes, yes.
Okay, thank you for your answer. Is there any feedback, Faisal Ma'ruf? If you don't want to give direct feedback, we are waiting your feedback in chat room. Okay. And then to Professor Seymour, there is a thumbs up for Professor Seymour from Ibu Budi from Unhas, University of Hasanuddin, and also questions from Ibu Triastuti Rahayu. There are 2 questions for Professor Seymour. The first is, my latest research focuses on biodiversity of bacteria and fungi in cemetery soils. The study is related to the potential risk of pathogenic microorganisms spreading from cemeteries to nearby residents, especially because cemeteries and residential areas are located very close to each other in Solo, Indonesia. Due to the limited funding, the number of samples that we can analyze is limited also. So how should we determine the sampling location and sample size so the collected sample can adequately represent the existing population? So this is maybe technical sampling method for eDNA research, Professor Mathew. And the second question is, can 16S rRNA gene be used to detect cephalopods in eDNA studies? There are 2 questions from Ibu Triastuti. Professor Matt, that is your time to answer the question.
Sure. Regarding the first question on technicality, it depends a bit on your research question. I would first probably look at designing how you want, what the explanatory variables are that you want to ask with regards to your urban and developed sites, and then figure out what the replication is that you need to ask it. If you're limited to a small number of samples, you need at least 3 replicates usually to differentiate between each explanatory variable. And so if you start adding in time or particular environmental variables, I don't know exactly what variables you're measuring in terms of trying to differentiate diversity, but just assuming if you have like a heavily developed versus low development site, if you want to have a gradient in there, make sure that you address enough replication in that. So keep it simple. If you have limited funding, that's perfectly fine. I think studies that prove proof of concept are highly valuable. But basically, you want to make sure you don't get caught in the situation where you're trying to over-test for limited data. But I think experimental design is a key factor here that you need to consider. And in terms of, what was the second question? What was the second question? I apologize.
Okay, second question is, can 16S rRNA gene can be used to detect cephalopods in eDNA studies?
Oh, yeah, okay, potentially, yes.
Okay.
They are a difficult one to get, and they probably don't, I have not seen studies that have successfully captured them. I guess their shedding might be interesting to deal with. I think I would probably try and do a pilot study first to test if these guys are shedding material adequately, either through artificially, probably an aquarium-based study. I probably would try and, usually that's what we end up doing if we can, is collaborating with some aquariums that have cephalopods, and then seeing if we can get measurements from them, because that eDNA from aquariums is highly condensed, and it should work pretty well with that situation. And then scaling it up to an actual live test, I think would be more what I would suggest.
Okay, thank you. That as far we know that 16S or RNA only can be used for microorganisms such as bacteria or archaea, and then now we know that also it can be used for cephalopods in eDNA studies. Really interesting. And then from Ibu Yuli Ayu from the University of Erlangga, or Erlangga University. Are you still here, Ibu Yuli Ayu? She says that That's really interesting, Mr. Matt. I'm also currently researching eDNA barcoding of crude oil. Okay, maybe you can further contact or collaborate with Professor Seymour. And then the next feedback is from Ibu Dewi Sahida from the National Research Organization of Indonesia. Ibu Dewi Sahida, are you still here? Okay. She writes that great research and presentation, Prof. Seymour. Do you have any collaboration project related to eDNA technique to detect the pathogens of fish or crustaceans? Okay, your time.
Not presently, no.
Okay. So presently, Professor Seymour doesn't have the project related to this topic, Ibu Dewi Sahida. Thank you, Professor Seymour. And then any other question from the participants? Okay. Oh, many chat in Q&A box. To Dr. Lutfi, how to validate or do a curation on the data submitted by the user of Peluru Taloka platform?
Thank you for the question.
Okay.
How to validate? We have two experts of botanists. First, after students capture the image, after that they review by the AI, generative AI, they submit to the platform, and it's not directly published. It will be a draft, and the draft will be reviewed by the botanist, the expert, about the taxonomy, about the medicinal plants and its benefit. And after review, if there something has to be clear, has to be fixed, the botanist will resend the draft to the students or teacher, and they have to resubmit it after revise the article. Yeah. After revise, and then the botanist feel that this article is proper, and it will be submitted after that. Thank you.
Okay, thank you, Dr. Lutfi. So basically the validation is based on the expert judgment, right?
Yes.
Okay, thank you. And then again for Dr. Lutfi, from Mr. Muji Santoso. Thank you, Dr. Lutfi, for highly comprehensive presentation. In your deep learning framework and workflow, you highlighted that taxonomic expertise is rare and expensive,
yes,
which lead to expert validation bottleneck. Yet your process flow shows that the entire decision support system for the herbal industry and researcher relies completely on database built from these expert verified labels. Since you also suggest that community voting as a potential solution to this bottleneck, how do you prevent community bias from compromising the data integrity required by the industry, and how will your deep learning model maintain its target of over 80% top first accuracy if the validation bottleneck forces a reliance on non-expert crowdsourced consensus?
Okay, I got it, the question. Thank you, Pak Muji Susanto, for your question. Actually, this framework and this workflow is, I stated it this is our vision, our future vision. But thank you for your suggestion that I have to, have to combine this solution, because, yeah, especially this is a benchmark from PlantNet, yeah, from PlantNet. In PlantNet, the community is not general community. This is a community with the botanist, with the ecologist inside, not general community, yeah, not general community, but the community is maybe association, maybe the association of botanists and ecologists, like that. So, and for the deep learning, this is our target, yeah, our target to maintain the 85% accuracy. This is still target, because we know that we are in the early stage. We have only limited resources, including the botanist expert. So we tend to ask help from AI, from deep learning, with database that trained and testing during our development of this application. So that's my answer. So this is still our vision, future vision. But thank you for your suggestion, Pak Muji Susanto. Yes, thank you, Pak.
Okay.
Okay, thank you.
Okay, thank you, Dr. Lutfi and Mr. Muji Santoso. So Peluru Taloka is still developing, and the target is deep learning and AI utilization. Okay, next we move to next question. I think many texts and already answered by Professor Mathew. And maybe I read once more for Professor Mathew from Ibu Dewi Sahida from the National Research Organization. Would you mind if I contact you sometimes by email to discuss potential collaboration research? Is it open for you, Professor Mathew?
Yes, of course. You're more than welcome to. Thank you.
Okay. Thank you, Professor. And then the question is for Professor Shafi. Has this application already used in clinical practice to support insulin administration for patients with diabetes? If so, could you please provide an example of its application? Professor Shafi?
Okay, thank you so much. Research in the biomedical field, like what I did now, for direct application to the patient is not such that easy. We have to have ethical clearance first. That ethical clearance must have the full enough evidence to show to the audience of medical field that our proposed solution is work well. And until now, because I don't have any collaboration with the medical field on this area for the diabetes system, so I don't have any direct application. So far, it's only based on the simulation. Okay. So again, if any in the forum from medical field, I'm open for the collaboration. We can do such, human test for the application. But before that, we have to go with the clearance, like I said, for the ethic clearance in medical field. Thank you so much for the question.
Thank you, Professor Seymour. Professor Shafi'i, I'm sorry. And it's already answered all questions, I think.
It is one question in the chat.
Okay.
Regarding interesting on my work, do you also perform the research on modeling in terms of renewable energy production? Yes.
Okay. Go ahead, Prof.
Yeah, we can think about to have the collaboration on the renewable energy production, especially when we involve the controller of optimization in that case. we can do such research together.
Okay.
This from Richard Christian Sanito. Thank you for the question. You can contact me via my email or via my phone number. No problem. Thank you so much.
Okay. Thank you so much, Professor Shafi'i, for open collaboration opportunity. And ladies and gentlemen, we have limited time to continue our discussion today. But before we close, we will deliver the certificate of appreciation. The first certificate is for Dr. Muhammad Lutfi Hidayat. Thank you very much, Doctor, and give applause for Dr. Muhammad Lutfi Hidayat. Committee, please share the e-certificate. Okay. The second e-certificate is for Professor Mathew Seymour. Here is it. Here it is. And thank you, Professor Mathew Seymour from the Hong Kong University. Thank you for sharing, and thank you for open collaboration for us. And then the third e-certificate is for Professor Shafi'i Samaun from the King Abdul Aziz University, Saudi Arabia. Thank you, Professor, for your sharing. It means a lot for us. Now, ladies and gentlemen, we have all the speaker session and Q&A session. Today's session has offered a rich exchange of ideas. We have explored how molecular technologies can support biodiversity conservation, especially eDNA, and then how digital competence and artificial intelligence can transfer education, and how intelligent system can offer new possibilities for healthcare and human well-being. This discussion reminds us that meaningful innovation requires more than technological advancements. It requires scientific integrity, ethical responsibility, collaboration across discipline, and strong commitment to improve society and protecting our environment. Once again, I would like to express our deepest gratitude to our distinguished speakers, Dr. Muhammad Lutfi Hidayat, Professor Mathew Seymour, and Professor Shafi'i Samaun. Thank you for generously sharing your knowledge, expertise, and inspiring perspectives with all participants. May this conference lead to new research collaboration, innovative educational practices, responsible technological development, and meaningful action for biodiversity, sustainability, and human well-being. Thank you very much for your participation and attention. Let's close this session. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Yasir Siddiq, Ph.D., for bringing us to a very interesting and productive session today. May all the presentation and discussion give us more insight on many emerging issues on natural science, environment, and its learning innovation. Now, I'd like to read some announcement for the participant. Please pay more attention. First, attention parallel session presenters. The parallel session will begin promptly at 1 p.m. Jakarta time. Please be in your assigned room a few minutes before the session starts. The Zoom link has been included in the abstract book. If you haven't submitted your presented slide, please send them to the committee as soon as possible. Second, for presenters whose papers are selected for publication in the Web of Science or WOS, information regarding the publication fee will be announced later. Please wait for the official announcement from the committee. Thank you. Saya ulang kembali. Pengumuman yang pertama, perhatian kepada seluruh pemakalah sidang paralel. Sidang akan dimulai tepat pukul 1 waktu Indonesia Barat. Mohon hadir beberapa menit sebelum sesi dimulai. Bagi yang belum mengirim file presentasi atau PowerPoint, mohon segera mengirimkannya kepada panitia. Yang kedua, bagi pemakalah yang lolos publikasi di Web of Science atau WOS, informasi mengenai biaya publikasi akan diumumkan kemudian. Mohon menunggu informasi dari panitia. Terima kasih. Before entering the last session, we would like to take the documentation from all the participants. Please turn your camera on and show your best pose. Okay, get ready. First slide. The second slide. Third slide. Fourth slide. Fifth slide. Sixth slide. Alhamdulillah, thanks to Allah, we finally finished all the agenda for today. On the behalf of the committee, we would like to say thank you for your active participation today. We ask for your forgiveness for any wrongdoing and inconvenience. Have a great rest of your day. See you in the 5th INCOBEST next year, Insyaallah. I am Gendis Anggista, signing off. Wassalamualaikum warahmatullahi wabarakatuh.